All right, welcome everybody. In this video, we are going to run a permutation test on a difference of proportion. So we've looked at permutation tests for a difference in means. And we're going to work with a data set that we've seen in class, which is data set that has information about various patients that had a melanoma lesion removed. And if you're not familiar with melanoma, melanoma is a form of skin cancer. It's one of the more common forms of skin cancer that also happens to be one of the more aggressive and deadly forms of skin cancer. And so the data set that we're going to be working with, if you want to read more about melanoma, there's a link up here. But the data that we'll be working with is in a package called boot, and the data set is called melanoma. So this first code cell is going to load the data, and we're going to get a summary of the variables in the data set. So we can see here, we've got, I believe, 7 different variables in the data set. And the ones that we're going to focus on in this video with our test in proportions is going to be the variable for sex and the variable for ulcer. And so the sex here we can see is initially being stored as a numeric variable, and that's because the classes of sex are 0 and 1. And so what we're going to need to do is convert the sex variable to a categorical variable before we do our analysis. And the same goes for the variable ulcer. So ulcer is telling us whether or not, so these are a bunch of patients that had a skin lesion removed that was melanoma. And sometimes these lesions are ulcerated, and that means that the skin breaks on top. And typically, ulcerated lesions means that they've evolved over a longer period of time. Well, ulcer initially is also being stored as a numeric variable. 0 means that the lesion that was removed was not ulcerated. 1 means that it was ulcerated. So we're also going to need to convert this variable to a factor. And so here, I'm going to run the structure command on melanoma, and we can see that initially all of these variables are being stored numerically. And so here I'm going to convert each of the 3 categorical variables in the data set to factors. We're not going to use the status variable here, but this is telling us whether or not the patient died during the study from melanoma, whether they survived, or whether they died during the study from other causes. But this is not going to be part of our analysis in this video. And so here we can see the breakdown by sex. There are 126 that are coded as 0 and 79 that are coded as 1. And we can also see that the ulcer variable was converted to a categorical variable, because now we're seeing counts that 115 patients did not have an ulcerated lesion removed, and 90 patients, the lesion that was removed was ulcerated. Okay, so let's say we want to do a test on a difference of proportions. And let's say we want to analyze the following statistical questions. And that is, are melanoma patients that are assigned female at birth less likely to have a lesion removed that is ulcerated compared to the melanoma patients that are assigned male at birth? So in this case, we have 2 variables. The explanatory variable is sex, and this is categorical. The response variable is whether or not the lesion that was removed was ulcerated, and that is also categorical. So when we're doing a test on a difference in 2 proportions, both the explanatory variable and the response variable are being considered as categorical variables. And so now when we set up our hypotheses below, the null hypothesis, recall, is that there is no difference in these 2 groups. So in this case, the proportion of removed lesions that were ulcerated was the same for male and female patients. So I'm going to let PF denote the population proportion out of all female melanoma patients that have a lesion removed. What proportion of all of those patients had a lesion that was ulcerated? PM is denoting the population parameter for the proportion of patients that were assigned male at birth that have a lesion removed, which is ulcerated. And so when we state this in terms of notation, the null hypothesis, recall, has an equal sign in it. So we're going to say the difference in the proportions PF minus PM is equal to 0. And the alternative here is these researchers are curious whether females are less likely to have an ulcerated lesion. So when we state the alternative hypothesis, that's what we want to claim. We're going to claim that the proportion of melanoma patients assigned female at birth with ulcerated tumors is less than the corresponding proportion of patients that were assigned male at birth with ulcerated tumors. And so stating that in terms of notation and population parameters, the alternative hypothesis would be PF minus PM is less than 0. Okay, so after we've formulated our statistical question and set up our hypotheses, the next step would be to collect a sample of data and calculate a corresponding test statistic from that data, which we're going to use to gauge which of the hypotheses seems more likely to be true. And so since our claims above in the hypotheses are in regards to a difference of population proportions, our test statistic is going to be the corresponding difference in sample proportion. So we're going to calculate the P hat F minus P hat M. And so in order to calculate these proportions separately for the sample of female patients compared to the sample of male patients that we have in our sample, we first are going to need to subset the data, since for now, everything is stored, male and female, in the same data frame, melanoma. In this first code cell, we are first going to subset the data into a female vector and a male vector, and then we'll use that to calculate the difference, the observed difference in sample proportions. So for the female data, we're going to go into the melanoma data set. We're going to select the females, and remember, those were the observations that had sex equal to 0. And this is a logical test, so that's why we have the double equal sign. We're going to select the variable ulcer, and we're going to store these values in a vector. So we're going to drop the data frame structure, and we're just going to create a vector of a bunch of values, 0s and 1s, that correspond to whether or not the lesion that was removed was ulcerated or not. So this last argument, drop equals true, is telling us to drop the headers and just store this data as a vector of values. And for the male sample, we're going to do the same thing, except now the males correspond to the observations that had a sex that was equal to 1. And we also want to create a pooled vector that contains all of the ulcer status for both the males and the females in the same data set. So that's just the original data set. So here I'm going to extract the ulcer variable from the melanoma data set, and I'm going to store this as the vector pooled sex. So this contains both the female and the male information about the ulcer status of the lesion. So now I would like to calculate the difference in sample proportions. So to calculate P hat sub F, we're going to go into the female sample, and this is a logical test. This is going to say, okay, is the value in this vector equal to 1? And if yes, we're going to assign it a numeric value of 1. And if it is not equal to 1, then we're going to assign it the numeric value of 0. And so this is going to count up how many observations in the female vector had an ulcer status of 1, and then it's going to divide that by how many total observations were in the female vector. So this mean female equals 1 is calculating the sample proportion for the females. This is doing the same calculation for the males. Okay, so although we're looking at functions in R that are telling me means, just keep in mind, we're applying this function mean to categorical data. So the result is actually going to be what proportion of all of the observations in each of these vectors were equal to 1, had an ulcer status equal to 1. So once I calculate these two proportions, I take the difference, and I'm going to store this difference in test diff prop. So I'm going to run the code cell here, and we can see that the observed difference here is approximately minus 0.1713. So this means that from our sample, we can see that the proportion of females that had ulcerated lesions were 17 percentage points less than the corresponding sample proportion of males that had a lesion removed, which was ulcerated. And so now with a hypothesis test, we consider this observed test statistic, and based on this statistic, does it seem more likely that the null hypothesis, there is no difference, is true, or that the alternative hypothesis that the females are less likely to have ulcerated lesions, whether that claim is true? And so just looking at this difference alone, we cannot be sure whether this difference could just be attributed to the randomness in sampling, or whether this difference is more significant beyond any difference that we would expect just due to the randomness in sampling. So that's what permutation distribution is going to help us measure. Okay, so question 2 is now asking us to create a permutation distribution. And so the permutation distribution is going to be our estimate. It's going to be a model for the null distribution. And the null distribution is the distribution of test statistics if we assume the null hypothesis is true. So in this case, our null hypothesis is that there is no difference in these proportions for females versus males. So we just group them all together in this pooled vector that we called pooled sex. And then we just are going to randomly assign all of these people in pooled sex to either a male group or a female group, just based on randomness alone and not based on their actual sex that was assigned at birth. So it's going to be useful for us to know how many observations we have in the original female data set, how many of those observations correspond to male patients, and how many observations do we have altogether when we combine both of those samples together. And that's what the length function is going to calculate in this code cell. So I can see that we have 126 females in our sample, 79 patients that were assigned male at birth, and then adding those two together, we get the total sample size, which is 205. So that's going to be important when we set up our permutation distribution. This code cell here is actually going to set up the permutation distribution. So this first line here, I just set the seed so that the output that I get is going to be consistent each time I run this. So my solution below is not going to change every time I run this code cell. So we're going to generate 10 to the 5th minus 1. So in other words, 99,999 permutation resamples. So we're not going to actually create every single possible permutation resample, but we're just going to generate a lot of them. And that's going to be enough to model the null distribution. And we're going to store each of our statistics after doing another permutation resample to a vector that I'm calling result.prop. And now we've got our for loop over here. And the first line of the for loop is going to tell us the index of the values from the pooled vector that we're going to assign to the female group. And therefore, the remaining values that are not assigned to the female permutation resample are therefore going to be assigned to the resample that we're considering as the male sample. So we're going to go in to N all. So this was the 205 values in our pooled sample. And out of those 2005 values, we're going to select the same number of values as we had in our original female sample. And here we're going to do this without replacing, since each person is going to go into one sample or the other. I don't want to pick the same person multiple times. The next line of code, sample.f, this is our permutation resample of females. So the index is telling us which values from the pooled vector are going to be sent to the female resample. And now this pooled.sex with index in brackets is actually now pulling off the corresponding ulceration status of those values in the pooled sex vector. And this command, pooled.sex with minus index in the square brackets, this is telling us to say all of the values that were not selected in index, let's take the remaining values and we're going to send them all to the permutation resample corresponding to the males. And then here we're going to run the same exact command that we did when we found the observed test statistic. Just instead of using the original data, we now use our permutation resamples. So this is calculating the proportion of the female resample that had ulcerated lesions, and this is calculating the sample proportion of the male resamples that had ulcerated lesions. And these two samples, sample.f and sample.m, are just being randomly generated. So they're not actually corresponding to the females and the males. We're just randomly sending a bunch of values, and we're going to consider them the female sample, and then we're going to send the rest of the values into the male sample. And now we're going to create a histogram to display those statistics. And I'm going to mark a red line at our observed test statistic, which was that minus 17%, or that minus 0.17 on our histogram with a red line. So this takes a little bit of time to run because we're going to do this 99,999 times. There we go. That took 7 seconds to run. And now I can see exactly where my observed test statistic lines up compared to all of the other statistics that we would get. So if there were no difference between the ulceration status of female patients and male patients, then this is what we should expect to get for the difference in sample proportions. But the one that we observed is way out over here. And so now we can see that this does seem pretty far removed from the differences that we should be getting if the null hypothesis is true. And so in order to measure just how extreme this observed difference of sample proportions was, we can calculate the p-value. And so the p-value is a conditional probability if the null hypothesis is true. So given that the null hypothesis is true, what's the probability of picking a sample, in this case of males and females, that have a difference in sample proportions as or even more extreme than the minus 0.17 that we observed? So that's the p-value, and that visually that is going to correspond to what proportion of the area, of the total area underneath this distribution, is sitting in the tail, in the left tail below the observed test statistic. So the next question is asking us to calculate the p-value. And for the p-value, we're going to first count up how many observations in our permutation distribution, and those values were stored in result.prop. So how many values in this vector in our permutation distribution are as or more extreme? So that in this case would be to the left, so less than or equal to the observed difference in sample proportions. And that's what we stored in test.diff.prop. So this is counting up all of the permutation statistics that are as or more extreme than our observed test statistic. But since our permutation distribution above did not generate all possible permutation resamples, it just did 99,999. There's no guarantee that we actually got the original sample as one of our permutation resamples. So we want to make sure that our p-value here is not zero, since we know that we do have at least one sample that is as or more extreme than the observed sample. So we want to add 1 to this total in the numerator to make sure that we have accounted for the observed sample that we got. Okay, because again, that observed sample might not be one of the permutation resamples that we plotted when we found our permutation distribution. So we include one more resample in our permutation distribution. That's the original sample. So that's why we add 1 to the numerator. So now we're considering 100,000 permutation resamples when we include the original sample back in with the other 99,999. And that additional sample that we include is the original sample. So its test statistic is equal to itself. So we also include it in the count in the numerator. And then we divide these two, and this is corresponding to the proportion of all permutation statistics that are as or more extreme than the observed test statistic. So in other words, that is corresponding to the area over here. So this over here is corresponding to about 1% of the total area underneath this graph. The p-value that we got from the code cell above is 0.0117. I just want to keep in mind that if you were running this code cell and you did not use the seed 3021, then you're going to get a slightly different permutation distribution, and that's going to result in a slightly different p-value from the one that I observed below, but they should be pretty close to each other. So in this case, the p-value is measuring how likely is the sample that we observed, given that the null hypothesis is true. So you want to be really careful here when you're interpreting p-values. We do not, and I stress, do not interpret this as how likely is it that the null hypothesis is true, for example. This is not calculating the probability that our hypotheses may or may not be true. We've assumed that our null hypothesis is true. Based on that assumption, how likely are we to get a test statistic as or more extreme than our observed test statistic? So this p-value is telling me that if there were no difference in the proportion of females that have ulcerated lesions and the proportion of males that have ulcerated lesions, then there would only be a 1.17% chance of getting a difference as or even more extreme than the one that we observed. So what's significant about that observation? So there's either two things that occurred here when we calculate this p-value. Either our sample was extremely unlikely, like a 1% chance, to be randomly selected from a population where there is no difference in the population proportions. So there is a 1% chance that this could have happened, but that is really rare and not that likely. The other possibility is that maybe our original assumption that the null hypothesis was true is incorrect. And so with the logical structure of hypothesis testing, when we have a p-value which is small enough, we haven't quite talked about what is small enough, but 1% is a really small chance, then we, instead of saying that the null hypothesis is true and we observed some miraculous sample, it's more likely that our original assumption in the null hypothesis was incorrect. And so therefore, our conclusion here would be to reject the null hypothesis. And we have evidence to support the claim in the alternative hypothesis. And therefore, we can claim that the proportion of melanoma patients assigned female at birth are indeed less likely to have a lesion that's ulcerated compared to melanoma patients that were assigned male at birth.